Previously, we looked at how individual alleles are sorted independently during meiosis thanks to homologous recombination. The probabilities of getting a specific allele combination and thus a specific trait can be visualized and calculated using a Punnett square. Each allele pair can be given its own Punnett square, with the probabilities of different conditions determined by the addition rule. We can use the multiplication rule to calculate probabilities between two different genes by multiplying each Punnett square probability together. Basically, each gene gets its own Punnett square with the two available alleles sorted as we saw before. We also looked at other ways that alleles can interact, including epistasis and incomplete dominance. Now, we want to look at another way that allele combinations can diverge from the more simple examples that we looked at before. The first of these are referred to as sex-linked genes. In humans, these genes exist on the X or Y chromosome. Let's look at how X and Y chromosomes sort during meiosis, and then we can look at how associated genes might work in a Punnett square. Humans generally have 46 chromosomes, or 23 chromosome pairs. These are broken up into 44 autosomes and 2 sex chromosomes. When these chromosomes are sorted into gametes, we can see that each gamete gets one sex chromosome, either X or Y, depending on what was present in the cell undergoing meiosis. Here we have a cell that starts out with 2 X chromosomes. During meiosis, each gamete will have one of these X chromosomes. In a cell that starts out with an X and Y chromosome, each gamete will get either an X or the Y. Eggs typically carry one X, and sperm generally carry either an X or a Y. We can see how this Punnett square would look. Sex chromosomes are unique in a few ways. First, even though the XY chromosome pairs will join up on the metaphase plate during meiosis, they don't generally experience homologous recombination of gene segments. However, XX chromosome pairs do participate in homologous recombination. This is because the X and Y chromosomes don't share homologous gene sequences, but the XX pairs do. This means that Y chromosomes are inherited directly and intact. It's amazing to think that Y chromosomes can be traced back to ancient ancestors who carried the same Y chromosome. For example, 13th century Mongolian warrior Genghis Khan's Y chromosome is present in nearly 16 million people living today. Just like autosomal chromosomes, X chromosomes can experience recombination and can have dominant and recessive alleles. However, in babies with one X and one Y chromosome, all of the traits on the X chromosome are expressed because there is no matching allele on the Y chromosome to interact with. The X chromosome contains many genes, both related to sexual characteristic development and those that are not. We can designate genes that are X-linked by using this superscript. Capital superscript means dominant and lowercase means recessive, as we have done before. We can make Punnett squares for X-linked genes just as any other gene. In this example, N is a gene that helps develop the ability to see color, and the recessive allele leads to color blindness. The Punnett square looks like this for a homozygous dominant XX organism and a recessive allele in the XY organism. Notice that none of the offspring will experience color blindness, but all of the XX offspring are carriers. However, if the XX organism is heterozygous for the recessive gene, then the potential offspring are shown here in this Punnett square. Half of all the offspring will be colorblind, but for different reasons. Notice that one of the colorblind offspring is homozygous for the recessive, and the other colorblind offspring has no second X chromosome, so the recessive allele shows through. The discovery of sex-linked alleles was made by Thomas Morgan, a researcher who looked at genetics in fruit flies. He found that most fruit flies had red eyes, but every once in a while, a fly was born with white eyes. When Morgan mated the red-eyed fly with the white-eyed fly, he got all red-eyed offspring in the F1 generation. This is what we would expect to see if the red-eyed allele was dominant and the white-eyed allele was recessive. Indeed, when he bred their offspring, the F2 generation showed a 3-to-1 ratio of red to white-eyed flies. This is consistent with the dominance pattern we have seen before. However, Morgan also noticed that all the white-eyed flies had an XY chromosome combination. He concluded that the gene for eye color in fruit flies was found on the X chromosome, and that flies with the XY combination didn't have a second X chromosome to balance the recessive white-eyed allele. So far, we have looked exclusively at alleles that follow the independent assortment rule. However, the reality is a little more complicated, because homologous recombination entails the swapping of sections of the chromosome. Sometimes genes are so close together that they will recombine together. This means they are linked genes, and do not follow independent assortment. Linked genes appear to sort during meiosis as if they were one gene. We can imagine a set of homologous chromosomes with two different genes on them, and these genes are close together. Because they are close together, when the chromosome recombines, it is more likely that they will travel together on the same segment of DNA. However, every once in a while, the DNA break that leads to recombination will occur between these genes, and they will be sorted independently. In fact, the distance between two genes on a single chromosome is related to how often they are separated by recombination. Genes from separate chromosomes are always sorted independently. But when they are on the same chromosome, they are sorted based on their probability of being split apart by homologous recombination. That probability goes down the closer they are to each other on the chromosome. Let's look at another test cross and see how Morgan identified linked genes. Morgan used a different combination that allowed him to track the sorting of alleles from just the egg. He looked at flies with combinations of two genes. One gene was for wing size, denoted by VG, for vestigial. It's important to note that this is still one gene, even if the abbreviation is two letters. The dominant wing size allele produces large wings, and the recessive produces small wings. The other gene was for body color, denoted by B for body. The body color dominant allele produces gray bodies, and the recessive produces black bodies. He crossed sperm that carried all recessive alleles, so a fly with small wings and a black body, with the egg that was heterozygous for both, as we normally see in the F1 generation. There are 4 possible outcomes of this cross. If these genes were independently assorted, we would expect to see a 1-to-1-to-1-to-1 result in the traits of the offspring as this chart shows. However, if the two genes were linked, we would expect to see 1-to-1-to-0-to-0, because the egg could not contain a recombined version of the genes. However, that is not exactly what Morgan found. Morgan's results were closer to 5-to-5-to-1-to-1. What does this mean? It means that the gene linkage is not 100%. Scientists can use ratios discovered in hybrid crosses like this to map out how close genes are on a single chromosome. Genes that are farther apart will have offspring ratios closer to independent assortment values, and those that are closer together will have ratios that are farther from independent assortment values. Homologous recombination provides sexually reproductive organisms with a massive increase in genetic diversity, and increases those organisms' ability to adapt to new environments. However, this also creates the potential for large-scale chromosomal alterations to occur. Major chromosome events are grouped into 4 categories: deletions, duplications, inversions, and translocations. In deletions, a section of a chromosome may be cut for recombination, but instead of recombining, it is lost. Duplications occur when a segment of one sister chromatid is cut and reattached to another chromosome. This would produce gametes like this. Notice that we have a duplication of these genes, but in another gamete we have a deletion of that segment. Inversions are similar, but instead the cut chromosome region will reattach to the same chromosome, but backwards. Gametes that have variant chromosome numbers are called aneuploidy. One of the most well-known examples would be Down syndrome. Down syndrome is the result of either different sorting during meiosis, or a deletion-duplication event where the long arm of chromosome 21 is duplicated to another chromosome, usually 14 or 15. Another chromosome variation is aneuploidy of sex chromosome. These are XXY, Klinefelter syndrome, which usually presents as smaller than average male sex organs, larger than average breasts, and often sterility. XYY, which occurs in about 1 in 1,000 males, is not associated with any variation in development. Similarly, women born with trisomy X tend to be slightly larger than average, but are not associated with any particular syndrome. While some of these variations are unfortunately treated negatively in society, they are really part of the natural variations in sexual chromosome sorting and crossing over. As we've seen in the past, large-scale chromosome changes like the fusion of human chromosome 2 are the product of these kinds of changes. Thus, These events are part of what can drive diversity, speciation, and evolutionary success.